I was just reading the latest clinical data on exercise and cognitive performance, and it changes how we should evaluate the link between movement and thinking. Exercise is described as good for the brain, but this claim collapses decades of molecular neuroscience into a single vague statement. The real story is far more specific. The link between physical activity and cognitive improvement operates through a specific protein called BDNF. Understanding this molecule and its function transforms exercise from a trend into a mechanistic intervention. BDNF stands for Brain-Derived Neurotrophic Factor. The term itself breaks down into its function. It is a protein produced by the brain that supports the growth and survival of neurons. Neurotrophic means growth supporting. This is not a general description. BDNF is a specific molecular signal that cells recognize, bind to, and respond to in predictable ways. The practical question is not whether exercise is good for you. The question is what specifically changes in the brain when you move, and whether that change is actually responsible for cognitive benefit. Most people understand exercise as a cardiovascular intervention, but the brain changes that follow aerobic activity operate through a distinct mechanism separate from cardiac benefits. When you exercise, your muscles increase their metabolic demand. They produce molecular signals that communicate with the brain. One critical signal is increased BDNF release. The brain does not produce BDNF in response to thinking hard. It produces BDNF in response to physical activity. This is a biological connection between body and mind at the molecular level. Voluntary exercise increases levels of brain-derived neurotrophic factor and other growth factors, stimulates neurogenesis, increases resistance to brain insult, and improves learning and mental performance. But this statement requires translation. Each of these benefits operates through specific mechanisms. Understanding which mechanism produces which outcome is essential for evaluating exercise protocols. The primary mechanism linking exercise to cognitive change is BDNF-dependent neuroplasticity. Understanding this connection requires understanding what BDNF actually does at the cellular level. BDNF binds to high-affinity receptors called TrkB, which are located on the surfaces of neurons. This binding initiates a molecular cascade that regulates cell survival, neurogenesis, synaptogenesis, and long-term potentiation. Each of these processes maintains a different aspect of brain function. Cell survival prevents neuronal death. Neurogenesis creates new neurons. Synaptogenesis builds new connections. Long-term potentiation strengthens existing connections. The supplied clinical evidence supports a narrower conclusion. BDNF elevation from exercise correlates with improved learning and memory performance, but the magnitude of the effect depends on exercise intensity and duration. BDNF is not exclusive to the hippocampus. This protein operates throughout the brain in regions that support learning, memory consolidation, executive function, and emotional regulation. This distributed location explains why exercise produces cognitive benefits that extend far beyond simple motor learning. BDNF affects the core systems for memory, judgment, and emotional stability. The discovery pathway to BDNF involved decades of neuroscience research. Scientists studying nerve growth factors gradually isolated and characterized the specific role of this protein in adult brain plasticity. This is crucial because BDNF is not a modern invention. It is an evolutionarily ancient molecule present in mammals, birds, and other vertebrates, indicating its fundamental importance to brain function. The conservation tells us something important. Exercise-induced BDNF elevation is not a luxury feature for optimized brains. It is a core survival mechanism that evolution has preserved for hundreds of millions of years. When you exercise, your muscles produce metabolites and signaling molecules that communicate with the brain. Some of these signals directly influence BDNF production in brain regions supporting learning and memory. The intensity and duration of exercise correlate with BDNF elevation magnitude. More intense exercise produces larger BDNF increases. Sustained exercise over weeks produces more sustained elevation. This is important because it means there is no threshold below which exercise produces no BDNF effect, and no point of saturation beyond which additional exercise produces no additional benefit. The relationship is continuous across a broad range. BDNF, upon binding to its high-affinity receptor TrkB, activates signaling cascades that regulate cell survival, neurogenesis, synaptogenesis, and long-term potentiation, processes that underlie memory consolidation and learning. Do not oversimplify the TrkB signaling cascade as a single pathway. Multiple downstream proteins are activated by TrkB, and these proteins initiate distinct cellular responses that operate in parallel. One key outcome is increased protein synthesis in neurons. BDNF TrkB signaling increases the machinery that neurons use to build new proteins needed for structural growth and synaptic function. Another critical outcome is dendritic spine formation. BDNF promotes the growth of dendritic spines, the small protrusions on dendrites that form the physical substrate of synaptic connections. More dendritic spines mean more potential synaptic connections. More connections create expanded networks with greater processing capacity and more complex informational integration. BDNF also directly influences synaptic strength through mechanisms of long-term potentiation, where repeated activation across the synapse produces durable increases in connection strength. Long-term potentiation is the cellular basis of memory. BDNF supports this process by promoting the molecular changes that make synapses stronger and more durable under activation. BDNF plays a critical role in the transformation of synaptic activity into long-term synaptic memories and is considered an instructive mediator of functional and structural plasticity in the central nervous system. This means BDNF is not just supporting growth. It is actively directing how experience shapes neural structure. It is the molecular mediator between what you do and what your brain becomes. When you look at the proposed exercise to cognition mechanism, the key limitation is that BDNF elevation does not automatically guarantee cognitive improvement. It creates the conditions for improvement. Exercise produces BDNF, but whether that BDNF-enhanced plasticity window is used to learn new skills or degrade unused skills depends on what you do during and after the exercise period. Do not claim BDNF is the sole factor in neuroplasticity. Multiple proteins, signaling pathways, and transcription factors all contribute. BDNF is critical and well-characterized, but it operates within a broader system. The important limitation is the exercise to BDNF translation. Most studies establishing this link used laboratory animals with controlled exercise access, controlled diets, and controlled stress environments. Translating these findings to human exercise recommendations requires acknowledging that chronic stress, sleep disruption, poor nutrition, and irregular exercise patterns can all blunt the BDNF response. Do not present exercise as an isolated intervention whose cognitive benefits can be predicted without accounting for sleep, stress management, and nutritional context. Individual variation in BDNF response to exercise is substantial. Genetic background influences baseline BDNF production. Age influences the magnitude of BDNF elevation. Training status influences adaptation. Some individuals carry genetic variants that influence BDNF production or TrkB receptor density, meaning they may achieve greater or lesser BDNF elevation from identical exercise compared to others. What does hold is the mechanisms. Exercise produces BDNF elevation in both laboratory animals and human study participants. BDNF binds to TrkB and triggers the molecular cascades that support neurogenesis and synaptic plasticity. What does not hold is any claim that a specific exercise protocol guarantees a specific magnitude of cognitive improvement without accounting for individual factors, lifestyle context, or learning engagement. So why does exercise actually help your brain? The answer is that movement triggers BDNF release in the brain structures that support learning and memory, and BDNF creates a plasticity window where new connections can form and strengthen. This mechanism evolved because in ancestral environments, physical activity was directly linked to survival challenges requiring cognitive adaptation. Brains that learned faster during periods of physical activity had adaptive advantages. In modern contexts, without survival threats, this same mechanism can be harnessed deliberately. Exercise creates the BDNF conditions that permit learning. The learning outcome depends on what skills you practice during that plasticity window. The cognitive benefit of exercise is most dramatic when you combine the BDNF elevation with active skill learning. Learning language while exercise-elevated BDNF is present is more effective than either intervention alone. This timing dependency is crucial. If you exercise but do not engage in meaningful learning during the BDNF window, the enhanced plasticity remains unused. Conversely, learning without BDNF elevation is possible, but proceeds more slowly. The magnitude of cognitive improvement also depends on exercise type. High-intensity interval training produces larger BDNF elevation than low-intensity steady exercise, but both produce measurable increases. Separate the mechanism from the outcome. The mechanism is well-characterized. Exercise produces BDNF. BDNF activates TrkB. TrkB initiates plasticity, supporting molecular cascades. The outcome depends on whether that plasticity is engaged in learning. Use generalized wellness language rarely when discussing BDNF. Instead, specify exercise type, intensity, duration, timing relative to learning, individual baseline factors, and stress or sleep context. The biological reality is that exercise is a powerful cognitive tool, but its effectiveness depends on understanding what is actually being modified at the molecular level and how that molecular change translates to behavioral outcomes. If you want to use exercise to enhance cognitive capacity, the priority is understanding that you have a specific window of enhanced neuroplasticity, and that window is only valuable if you use it for meaningful learning. Match the exercise intensity to your recovery capacity. High-intensity exercise produces greater BDNF elevation, but also greater systemic stress. In chronic stress or sleep-deprived states, this can be counterproductive. Measure what is actually supported, not how you feel after exercise, but whether your ability to acquire new skills and consolidate memories has improved during training periods. Recognize that BDNF elevation creates potential, not guarantee. Potential for learning, for memory consolidation, for new connection formation. Whether that potential becomes actual improvement depends on your behavior during the plasticity window. This episode is educational interpretation of established neuroscience about BDNF, TrkB signaling, and neuroplasticity. It is not medical advice. Significant changes to exercise routines warrant discussion with qualified professionals. The goal is understanding the BDNF mechanism accurately enough to evaluate exercise claims with appropriate expectations and enough nuance to distinguish molecular mechanism from behavioral outcome. Cognitive capacity is not determined solely by BDNF levels. BDNF supports the biological conditions for learning, but cognition is shaped by what you practice, how consistently you engage, and what challenges you pursue. The BDNF pathway is a well-characterized biological mechanism that links exercise to cognitive improvement. That precision deserves a conversation that matches it in specificity and avoids oversimplification. The biology shows clearly that exercise is a cognitive tool, but tools require proper use. Understand what BDNF actually does. Time your learning with your exercise and engage meaningfully with the plasticity window that movement provides. Optimize the protocol, protect your brain, and I'll see you in the next data set.